Hi guys, welcome to the Revive Stronger podcast. I'm your host as always, Steve Hall, and I'm joined here by Alex KT. I'm going to say KT and let Alex say the pronunciation of his last name correctly because it's one of those that I think I will mess up.
Yes, Alex Kolliari-Turner.
Awesome. So, Alex, you may have heard from Danny Lennon at the Sigma Nutrition podcast, which again is a fantastic podcast. And Alex went over some brilliant things there. If you didn't hear that, awesome. We've reached some new people and we're kind of highlighting something that's going to be really helpful to many of the listeners, super interesting as well. And so Alex is a second year PhD student at the University of Brighton. And he is currently conducting research into the effect of anabolic steroids on muscle gene expression, epigenetic markers, and their influence on the enhancement of muscle memory. And this is some of the things we're going to dig into. And first of all, I wanted to kind of hand it over to Alex to kind of talk about kind of what got him interested in this, or first of all, kind of why he's on the podcast, of like what has driven him to me initially, and somewhere where lots of the listeners will be able to potentially help. And we really hope we can help him because we're all interested in science and furthering the field. So if we can help Alex and help that mission, then that'd be brilliant. And then Alex, if you want to delve into kind of what got you interested into this particular area of research as well, that'd be awesome.
Yes, so I'm a second year PhD student at the University of Brighton, and research is currently looking to recruit 4 groups of participants. The first group is sedentary males that do not lift weights to act as a control group. And then we want to recruit males who lift weights regularly for roughly 8 hours a week, and they do not use anabolic steroids. And then we're looking to recruit males who lift weights regularly and they are currently using anabolic steroids. And when they come to our laboratories for sampling, they will be concluding a steroid cycle soon before they get sampled. And ideally, they've been taking steroids on a cycle for roughly 20 weeks. And then at that point, we sample them. And then they then go off anabolic steroids for 20 weeks and they would conduct a PCT. And then we will resample them 20 weeks later, with the idea being to investigate how much muscle mass they have lost at that time, and also how their muscle fibers have changed at the molecular level and anabolic pathways at the gene expression level, but we'll touch on that later. And then the last group, we're looking to recruit guys who used to take anabolic steroids 2 to 4 years ago, but since then have been drug-free or have been on a genuine TRT dosage of testosterone and have the bloods to prove that they have indeed only been at the physiological level since that point. We can also consider the recruitment of them. Everyone's got to be a male, age 20 to 35. And sampling is undergoing at the moment. We have sample dates on Saturday the 16th and Sunday the 17th of March 2019, and Saturday 13th, Sunday the 14th, and Monday the 15th of April 2019. And that's why Danny Lennon invited me onto his podcast, episode 270. And I'm here now with Steve because I want to spread the message of this research that we're doing, because it's very interesting for a lot of people. And I'm also in the need of finding participants as we speak.
And if they want to reach out to you, Alex, we have your email address below. Is that the best way to get hold of you?
Yeah, so my email address, my academic email below is the best way. If you want to find out more and be able to share the details of the study that we're about to describe and the purpose of why we're doing this, I have some posts on social media that can be easily shared. If you just search Alex KT and muscle memory into Facebook, you'll find a public post I've made that can easily be shared. Also Alex_K_T on Instagram, and you'll find a public account and post I've made that can easily be shared on there. And Greg Knuckles recently shared it on his Stronger by Science page, and Mike Isotel shared that on his Facebook page as well. So there's multiple ways to find the advertisement and a little bit of details about the project. But definitely, if you are interested, email me. And if you're eligible and you fit into those criteria and you're between 20 to 35, I'll basically organize a phone call and get in touch, because we reimburse everybody's travel expenses, and we'll even think about it if you're from Europe to do that, because
Amazing.
flights nowadays are pretty damn cheap, especially if you are a past or current user of anabolic steroids, as that's the most important groups, and obviously the groups hardest to find, as there's less of those guys out there, and especially because we have to get them in that magic moment where they happen to be cycling off anabolic steroids. And a lot of the guys we've got actually are happening to go off anyway, like they've completed maybe powerlifting competitions, and they're planning on taking a break after the powerlifting competition and going off cycle, and they want to just see how much muscle mass they're going to lose during that time, and they contribute to our greater understanding of how anabolic steroids increase muscle growth and potentially enhance muscle memory as time passes. And a few other guys are just cycling off because they want to take a break, and a few other guys are cycling off because they actually want to get their wives pregnant, and so it's sort of fitting in with their general schedule in their cycle plans anyway. And even if you can't make those 2 dates in March and 3 in April, we do it all throughout the year. And so do get in touch. Maybe you might end up being able to coincide your steroid cycle conclusion and you're going on a PCT anyway with our research, and then just greater benefit the scientific knowledge, and at the same time find some information about yourself, because we'll measure your muscle mass with a bioimpedance scale, research grade skin fold calipers for your body fat percentage, and do a bod pod reading. And you've got to normally pay a few £100 for a bod pod reading, whereas at least this, obviously you get it for free and you get to come down to our labs, all travel expenses paid. So that's the benefit. Well, unfortunately, we can't pay anyone. That's the university's like ethical rules with recruiting participants. If there can't be money exchanging hands, but we can reimburse your travel expenses, provide you with food, and obviously any of the muscle measurements you get are complimentary in that regard.
Awesome. I mean, it sounds like a great opportunity for people if there are, and I mean, we have hopefully a decent listener base, and there are hopefully some people who are at that point. I mean, if we're reaching people across Europe, and I know we have loads of coaches that listen to this, and if anyone wants to kind of give a favor to the podcast and to just science in general, share it around, because you're coaches and you probably have people following you and can kind of reach out to this wider audience. So that'd be fantastic. Question.
And it's been the best way of finding people at the moment. And I would just say one thing while I'm talking about the logistics of the study is obviously everything is anonymized. So nobody knows who's taken part. You're given a random identification number, and we don't discuss who's taken part. There's no details. And as soon as we do the initial paperwork and consent, you're just anonymized from that point onwards. And that is taken very seriously, especially because we're under ethical obligation to do so. And so there's no concerns in that regard.
Fantastic. And out of interest, and I think the listeners will be interested, what got you into this arena of kind of research? What specifically about this is interesting to you, Alex?
So I did an undergraduate biology degree, and then I took a brief break from that and got involved with some teaching, and then wanted to go back into academia. And I was living in Brighton at the time, and I touched base with my professor that's based out of there now, and he was involved in anti-doping at the time, and still is involved in anti-doping, and he was trying to generate new drug tests for the drug erythropoietin, or EPO, which is a drug commonly used by endurance athletes to increase the amount of red blood cells they have to increase their aerobic capacity and oxygen carrying capacity of the blood. And he's trying to develop a new drug test by looking at changes in gene expression in whole blood, as opposed to just changing metrics of red blood cell fluctuations and immature red blood cell fluctuations through time, which is what the standard athlete biological passport uses at the moment to try and catch people who do blood doping. And I wanted to get involved in helping in that project. And then at the time when I started to get and because I really do think that sequencing and next generation sequencing and understanding our DNA and our RNA and our transcriptome is a fantastic and a very interesting piece of science that is understudied at the moment because the technology is so new and now becoming quite affordable. And so to be able to apply the omic technologies, transcriptomics, metabolomics, proteomics to anti-doping is possibly the way the field is going to move forward, as opposed to looking for presence and absence of the drug in urine or fluctuations in blood parameters through the blood tests. And the omic technologies are going to potentially, via research, if we find interesting things, provide new biomarkers. That's what he was interested in with the EPO. Question, and so he was trying to find new biomarkers with changes in gene expression with transcriptomics in whole blood, and he did a dose administration study with EPO and double-blind, placebo-controlled, and also measured their increases in performance, and as actually found that there are some genes that are changed after EPO has been introduced into the system, and they become upregulated and then downregulated, and that signal of downregulation can last a little bit longer than the fluctuation in red blood cells. And so I found that whole avenue of research very, very interesting. And when I got touching base with him, he basically said that he was in the process of organizing this research project, and it sort of went from there.
Awesome. No, it sounds incredibly interesting. And I think it probably warrants quite a bit of background. A lot of the listeners will probably recognize some of the things you're about to talk about. But it might just really help understand fully where this research is going, what you're trying to truly identify. And I don't know if you want to start off with kind of some of the processes of hypertrophy. Many people will have heard about kind of incorporating myonuclei, but if you want to talk about that kind of background, and then that will lead into kind of where the study will go and some things that we hope you might find out.
So muscle fibers are very interesting cells in the human body because by volume, they are the largest cells inside the human body. So, for example, the sartorius muscle, which goes across your quadricep, can be 23 inches long, that cell, that muscle fiber. And so it proposes a diffusion problem for the cell because, say, for example, a liver cell has one nucleus per cellular body. Whereas for a muscle fiber to be able to transport proteins across its whole cellular volume, which is a huge distance, you're not going to be able to achieve that with one nucleus inside the cellular body. So you have to be multinucleated. And so muscle fibers are among a very small subset of cells inside the human body that are multinucleated. And the fancy term for that is it exists in the syncytium. And these nuclei are responsible eventually for the production of the proteins inside the muscle fiber. And obviously muscle fibers, you can imagine them as these long cylindrical cells that contain contractile proteins that slide past each other to result in muscle contraction. And so these nuclei are responsible for making those contractile proteins. And with hypertrophy, the name of the game is to try and have protein synthesis to outweigh protein breakdown consistently through time. And if that happens, muscle mass is accrued in terms of the muscle contains more proteins and the fibers as cylinders expand in size and their cross-sectional area gets bigger. Now, there's two ways in which that can occur. The existing myonuclei that are already inside the muscle fiber can make more proteins, and then the cylinder will expand in size because the more proteins are being made. However, there is only a fixed distance in which a single nucleus can have proteins diffuse away from it, and that's the so-called myonuclear domain. And it seems to be that there's actually a ceiling to growth in which if you manage to get all of the nuclei inside the muscle fiber to maximally produce all of the proteins they possibly could, and the diffusion distance was absolutely maximized, the fiber would only be able to get to a certain size. And for it to surpass that size, the only way it's going to happen because of this diffusion distance problem is for more nuclei to get incorporated into the fiber. And the interesting thing about the nuclei inside muscle fibers is they're post-mitotic, which is just a fancy way of saying they can't divide by themselves to make new nuclei. The only way you can get more nuclei into the fiber is via the activation of the stem cells that surround the muscle fiber, these so-called satellite cells, and to cause them to put their nucleus inside the muscle fiber. And the satellite cell will actually divide in two. One cell will remain as a satellite cell so that you don't deplete your stem cell pool, and the other cell will donate its nucleus into the muscle fiber for it to become a true myonuclei. Now, the interesting thing is that process happens through natural hard training, and people and studies have demonstrated that in individuals, their muscle fibers in humans will increase in size up to a certain point without any myonuclei donation. And then if they want to get even any bigger, they have to incorporate myonuclei donation. It is possible, and there are studies in mice where they have basically genetically modified the satellite cells so that when you give the mice a drug, it kills all the satellite cells inside its muscle. And so there can be no myonuclei donation occurring, and muscle growth actually still occurs in those mice. So it is possible. And so what seems to be occurring is that there is sort of it depends at the rate. It's the limit of how many proteins is the myonucleus currently making. If it's working at its absolute maximal output, then there's no more propensity for that muscle fiber to get any bigger. The only way it's going to get bigger is by more myonuclei going in. But if the myonucleus is not working at its full maximal output and has the potential to make more proteins, then that fiber just can continuously expand in size. And that's what we see in these mice where satellite cells are genetically destroyed. So that's sort of the mechanism of muscle growth. But what's become very, very interesting is that studies in mice have demonstrated that if you expose the mice to a hypertrophic stimulus, and so you increase their myonuclei number and their muscle fibers get bigger, but then you expose them to an atrophying muscle stimulus, so you want the muscle mass to decrease in the mice, and they do that in numerous ways. They do that by suspending their hind limbs, so they prop their legs up, and so the legs are not contracting at all. So if there's no muscular contraction, there's less stimulation of muscle protein synthesis. Muscle protein breakdown outweighs muscle protein synthesis through time. Muscle atrophy occurs. You could also cut the nervous supply physically to the muscle. Again, no muscle contraction, no muscle protein synthesis. Breakdown outweighs it. Muscle goes down. You can also chemically inhibit the nervous supply, and that also induces muscle atrophy. But in a groundbreaking study that was conducted over a decade ago now by a group of researchers in the University of Oslo, where they monitored mice after muscle hypertrophy have occurred and during all of those different atrophying conditions I just mentioned. And they looked at the exact same muscle fiber throughout that whole timeline. They found the muscle fiber got bigger in size and more nuclei. And then as the atrophying condition went on through time, so 7 days and 14 days, the muscle fiber shrank. As a cylinder, it got smaller, which you'd expect because its protein content is getting smaller. So those proteins are getting degraded. But the nuclei that were incorporated inside that, the true myonuclei, did not decrease through time, and they remained. And the hypothesis goes that those nuclei that are incorporated and remain will then facilitate muscle regrowth in the future, because all you have to do is reactivate those myonuclei to make more proteins, and the fiber will increase in size quicker in that second exposure to a hypertrophic stimulus than the first time exposure when you have to incorporate them in the first place. And so you can imagine that this is the mechanism now of how muscle memory occurs. Mm-hmm. So if you are an athlete and you train hard and you get, say big quads, you break your leg, it's a tragic accident, it goes in a cast and you lose that muscle size, there's no contraction, you could then put on quad muscle mass quicker the second time round than the first time round, because any residual myonuclei that were incorporated from your first growth phase are then remaining through time, and then they help in enhancing muscle mass later down the line. But the interesting thing is, resistance training by itself incorporates myonuclei, mm-hmm. and studies in humans where they have given them injectable testosterone, so anabolic steroids, and they have also not subjected them to resistance training, also results in myonuclei donation. So, and in mice as well, we know that just giving them steroids results in myonuclei donation. And that's because anabolic steroids work by binding to the androgen receptor. And the androgen receptor is present within many different cell types in the human body. And that's one reason why, although people take anabolic steroids to result in muscle growth, the androgen receptor is present inside the prostate, inside the brain, inside the skin, and binding and androgen receptor binding in those cell types can result in some of the adverse side effects that people don't want when they take anabolic steroids. And once anabolic steroids bind to the androgen receptor in satellite cells, they can cause that differentiation of the satellite cell and cause nuclei donation to occur inside the muscle fiber. And at the same time, anabolic steroids can also stimulate protein synthesis to be higher. So any residual nuclei that are already in there are going to be receiving more signals to make more proteins, and the steroids are then causing more nuclei to get incorporated as well. So you've got that double whammy to increase muscle growth, which is a good thing if for those that are doing that for that reason. And there was a study that was conducted in 2013 by that same group, Christian Gunderson's lab. The lead author in this one was Ingrid Egner, published in 2013, where they exposed mice to anabolic steroids, and they saw an increase in muscle growth. The cylinders, the muscle fibers got bigger in size, and the nuclei content went up. And then they exposed them to 3 months of atrophy. So they removed the steroids that were only incorporated for 2 weeks. And 3 months later, the muscle fibers of both groups, so the group that was exposed to steroids and the group that wasn't, decreased in size. And they actually ended up being at the exact same size. But the nuclei numbers were elevated in the group that were previously exposed to anabolic steroids. And then when they exposed that group of mice to a hypertrophic stimulus, it was actually a synergist ablation, which is where they cut one muscle off and the mice is forced to walk around on another muscle, and it causes massive muscle growth. Although that is a little bit of a damaging surgical procedure, and that's potentially one negative of that study, it still is a sort of, I guess you could say, industry standard of how to induce muscle growth in the mice very quickly, because the force of it having to walk around on that muscle the whole time causes a large amount of muscle growth. But irrespective of that fact, muscle growth, the rate was much, much higher in the group that was previously exposed to anabolic steroids, and it's deemed to be because they had higher myonuclei numbers to get go when they were exposed to that resistance to synergist ablation at that time. And the rate of muscle growth was about 30% higher over a 6-day period, and muscle did grow in the other group, of course, but at the end of that exposure to the hypertrophic stimulus, the muscle growth, the total muscle size fibers was actually 20% higher in the group previously exposed to steroids than the other group. And so that lead author of that laboratory, Christian Gunderson, has now come up with this theory that upon first training, myonuclei are incorporated into a muscle fiber, and those nuclei then make more proteins. The muscle fiber cylindrically expands. If it's then exposed to an atrophying condition, because there's less stimulation of protein synthesis, protein breakdown outweighs protein synthesis, the muscle fiber as a cylinder shrinks, but any nuclei that were incorporated are remained. And then if they're then exposed again to a hypertrophic stimulus, they just switch on from protein synthesis at a faster rate, and second growth is indeed much quicker. And so the question now is, do the nuclei that incorporate from anabolic steroid usage in humans linger for a significant period of time? And so in that mice study, that was 10% of the mice's lifespan, which the nuclei remained. And we essentially have next to no research on myonuclei decay rates in humans. I'll read a little section here on Gunderson's paper where they say, there's only really been in his review paper in 2016 where he says, we have demonstrated that in mice, the memory effect lasts for greater than 10% of the animal's lifespan. In humans, the turnover of cells has been studied by utilizing the peak in carbon-14 availability after post-war atmospheric testing of nuclear bombs. Intercostal muscle tissue was harvested from 2 individuals aged 37 to 38, and the carbon-14 content of their genomic DNA indicated an average age of their nuclei to be 15.1 years. And that's the only evidence that they mentioned in that paper of the potential lifespan of a human myonuclei. And there's flaws with that. I mean, that's an intercostal muscle. That's a muscle in between your ribs. And that was only from 2 people, aged 37 to 38. So we don't know at what point that myonucleus was incorporated, and if that is an accurate representation. And it's obviously very, very difficult science to be able to do. And so our study is basically finding guys soon after they've finished an anabolic steroid cycle, because people are taking these drugs to increase their muscle growth. So they should at that point have a large amount of muscle mass, and nuclei incorporation should have occurred. And then the question is, when they cycle off 20 weeks later, what happens to their nuclei number and how much muscle mass did they lose? And that's why we are then sampling at that 20-week point. And then the guys that took steroids 2 to 4 years ago, are their myonuclei numbers elevated? Yes or no? And there's only been one pretty big study that was conducted in 2006 by this guy called Anders Ericsson at the University of Umeå in Sweden. And he recruited exactly the same groups that we're looking for. And he biopsied their vastus lateralis, so their outer quad, and their trapezius muscle. And he found that in individuals who used to take anabolic steroids in their trapezius muscle, they had elevated numbers of myonuclei, and they were retaining this high number. And so that's potentially some indirect evidence that the nuclei that incorporated from steroid usage in humans are not going down through time. But again, that was an observational study. And again, ours is an observational study because of the difficulty of getting the ethical passing to give people anabolic steroids. And that study is also that same cohort of people in the year 2000 when they initially did some initial research on just controls, non-steroid using people and steroid users. That was the study that found out that the androgen receptor density is much higher in the trapezius muscle for guys that are on steroids than guys that are not. And even on people who do not take steroids, the androgen receptor density is higher in the trapezius muscle compared to your vastus lateralis. And then the guys that take steroids, that discrepancy just goes up slightly. So the vastus lateralis has less than the trapezius, much more, and even more than the clean guys. And so that anecdotally kind of makes some sense because, guys that have incredibly large traps, you tend to see them in the enhanced realms of bodybuilding and not so much in the non-enhanced realms. But that's an anecdotal observation. So the muscle of our choice is the trapezius muscle, and that's why we're deciding to look at that, because it has been shown to have high numbers of myonuclei in past steroid users. So if there's a muscle to investigate the muscle memory effect, it would make sense to pick that one, and especially if its androgen receptor content is higher than the other muscles. So there's been past research on what are the nuclei numbers like in current and past steroid users, and I actually spoke to a guy from the University of Denmark who has basically recruited the same groups that we have without sedentary controls, and they've got their vastus lateralis, and they are, as we speak right now in March, starting to analyze that data and work out what are the nuclei numbers like. Again, observational study, but still will add to our knowledge. But the unique thing about our research is that we are storing the muscle inside a genetic preservant. And so we're going to have the ability to store both muscle DNA, so the myonuclei DNA, and also the transcriptome, so all of the transcripts inside the muscle. And that means we'll get a snapshot of what is the gene expression like inside that muscle. And that will give us an idea of what anabolic pathways at the molecular level are being upregulated because of anabolic steroids compared to people who don't take them. Because anabolic steroids, when they bind to the androgen receptor inside muscle fibers, they move into the nucleus of the muscle fiber. They move into that myonuclei. They bind to very specific regions of DNA that are actually called androgen response elements. And then that creates a transcript that is eventually converted into a protein. And that protein then causes the differences we observe inside the muscle. And the question is, well, what transcripts are upregulated inside human muscle after it's exposed to anabolic steroids very recently, 20 weeks later, and then years later? Is there any differences going on? And that will better our understanding of how anabolic steroids just cause muscle growth, full stop. And at the same time, we have the ability to preserve muscle DNA. So we're going to be able to see the markers on the DNA, the epigenetic markers, and see what regions of the DNA are opened or closed inside the muscle fibers. And maybe certain areas of the DNA are more open in people that have taken steroids that are causative for more anabolic pathways being expressed, and compared to guys that don't take steroids. So that's never been done before. So that is just going to enhance our knowledge of how steroids cause muscle growth, which is interesting for everyone, really. Mm-hmm. So that's kind of the unique spin on our research compared to the standard myonuclei counting that's been done before, albeit there's not many studies like that out there. Yeah. That's pretty much everything that we're sort of aiming to do. And I think the really cool thing is we'll be able to have this sort of like timeline of someone who's just finished a cycle, and then see, does their muscle mass maintain, does it decline? And the same with the anabolic pathways inside the muscle. Does it go down? Does it not? Because steroids, they're all based off testosterone and all those different derivatives. So trenbolone, nandrolone, the oral steroids, turinabol, dianabol, whatever, they all have structural derivatives of testosterone or nandrolone, and they interact differently with the androgen receptor. So there could be differences based on what steroid you're taking. Yeah. And what is really interesting is in 2018, there was a paper published by a researcher called Michael Rossetti. I'll just get the title up, and it was called Androgens Induce Growth of Limbs, Skeletal Muscles in a Rapamycin Insensitive Manner. Lead author Michael Rossetti, Florida State University, 2018. And essentially what they did in that study in mice is they inhibited mTOR. And mTOR is the master regulator of protein synthesis inside the cell. You need to activate mTOR to be able to activate muscle protein synthesis. And that's why the recommendations are to eat 30 to 40 grams of protein for most guys, 4+ times a day with over 3 grams of leucine in it to maximally stimulate mTOR. And that's regarded as sort of the main master regulator of protein synthesis inside the muscle fiber. But you can inhibit mTOR in mice chemically with rapamycin, and that's what they did. And they then gave those mice nandrolone decanoate, an anabolic steroid, and those mice still experienced muscle growth, which is kind of crazy. So that means there's a redundancy potential in the system to cause muscle growth that anabolic steroids somehow have the ability to activate that is not there without them being there. So that is a bit of an eye-opener, and I'm sure there's groups out there as we speak that are further investigating that in mice, because that's really interesting just to understand how muscle growth happens full stop. Because if there is a redundant system to mTOR, we need to know what that is, just a better understanding of muscle growth for diseased populations, right, for sarcopenic populations, for the people that have got muscle wasting diseases, cancer muscle wasting diseases, HIV muscle wasting, and just the decline in muscle mass through age. Because the problem is as we age, our stem cell count and our stem cell pool decreases. So the amount of satellite cells in elderly people is way less than the amount of satellite cells in young people, and their testosterone's going down. So they've got less circulating anabolic hormones, and they have less satellite cells. So their propensity to build muscle just goes down with time. So muscle memory links into this, because it might be the recommendation, and Gunderson says this in a lot of his papers, that public health for governments might say you should actually encourage people to weight train when they're younger. They've got more satellite cells, so myonuclei incorporation is more likely, and more testosterone. And if they can build up a certain level of muscle mass, they might be able to retain those myonuclei later through life. And then when they become older and myonuclei incorporation is just statistically more unlikely, they're still there hanging about and can help them maintain a high level of muscle mass, and therefore decrease the likelihood of being frail and and increasing the likelihood that they have their own mobility and they don't fall over and have a hip fracture and things along those lines, which can be obviously devastating for someone in old age and make them bedridden. So it may well be that the encouragement should be that people should be lifting weights to help them maintain muscle mass in later life, or help them have that ability to maintain more muscle mass in later life if their myonuclei numbers are elevated. But I would say as well, just about this myonuclei number thing, is that even if anabolic steroid users in the past are maintaining high numbers of myonuclei, that only gives them, at the moment, from my understanding, a theoretical advantage, because it should mean that they have a enhanced protein synthetic rate inside their muscle, because compared to someone who has less myonuclei. But they still need to do all those good things of the hard training and consistent eating and all the other lifestyle factors that come along with it to actually witness the benefit of having those high numbers of myonuclei. Because if they're not, then nothing else is sort of happening. It's not like you could go on a steroid cycle one time, train like a maniac, go off them, do absolute FA, and then come back to training and get to the same size you were before with a subpar training program, sometimes high nutrition. No, it's not going to work like that. Okay. The extent of the advantage is something that we don't know. Okay. It'd be very, very difficult. And the only way we're truly going to know that is via a administration study where we give anabolic steroids to people, put them on a training program, take them off anabolic steroids and off training, and leave them for months, and then get them back to training without steroids in their system, and then see how much muscle do they grow and their rate of muscle growth at that point, their myonuclei numbers at that point, their gym training performances at that point, and then we'll get a better understanding of what's going on. Because our study is only observational. We're only looking at these people, and I've got no control over the training or the nutrition of the steroid users once they cycle off. And if you're diligent and careful and you are dedicated with your nutrition and training as you cycle off anabolic steroids, obviously you're going to retain more muscle mass than somebody that does not do those things. And that could be a confounding factor, because myonuclei numbers in that group 20 weeks later could remain elevated, but they could be remaining elevated because they're all diligent with their training, and therefore their natural training that they're doing at that time is not making an atrophying condition that we could observe myonuclei loss occurring with. So that's a big confounding factor with the study that we're doing. And so the conclusions that we can draw from this are limited, but it's very much going to open the door to try and push for further research in this area. And at the same time, better our understanding with the gene expression and epigenetic markers of anabolic steroid usage. And at the same time, better our understanding of the decline after a cycle, because that's never been done before. But still, we have the constraints of it being an observational study due to the ethical nature of trying to give guys steroids. So, especially in this realm of research, there's going to be flaws because of that constraint. But I mean, this will open the doors to maybe more research, maybe with other drugs, and maybe or maybe it would encourage the areas of research where they do give people anabolic steroids, so the muscle wasting diseases, the cancer, the HIV muscle wasting, etc., to then monitor those people if they ever then go off steroids, to then see what happens, because there's a more of an ethical reason to give them the steroids. But then the problem with that is they're a diseased population, and so there could be a confounding factors there. So you'd want to do it in healthy people, but is that ever going to happen? We don't know. So, there's only limited things we can draw from this, but I'm excited by it because of the things that we will learn, and from the avenues that will open up and just enhance our knowledge and better understand myonuclei decay rates, but also better understand the transcripts that are upregulated in muscle post-steroid usage and the epigenetic changes inside the DNA.
Amazing, Alex. I think probably a lot of listeners' minds are a little bit blown, and mine included. So much information covered there, and truly kind of incredibly interesting in terms of like the implications of steroids for hypertrophy, in terms of like more myonuclei domains available to then hypertrophy. And I guess it's a case of kind of, we've seen anecdotally this muscle memory effect. Does that myonuclei domain remain for someone who's incorporated via previous steroid usage or anabolic usage? And it very much, it's funny, it reminded me a lot of kind of fat cells, and the fact that kind of I presume this is still the correct theory, in that you kind of, if you get new fat cells, they're ones that remain and you can't kind of get rid of them. They're then available to then potentially regain fat, potentially a little bit easier. So previously obese people can more so gain fat. I don't know if that's something that you've kind of noticed yourself, being a reflection.
Yes, I mean, Gunderson in his papers talks about how fundamentally we're looking at a cellular memory system here, which is just interesting as a biological subject, because everything has got to be looked through the lens of evolution. Why is there a memory mechanism inside the muscle that it remembers being previously large? And he suggests in his papers that this mechanism occurs in muscle fibers because it's beneficial on an evolutionary timescale. Because if you were to incorporate muscle mass seasonally, because back in the day when we're hunter gatherers and food availability is good, and you're hunting and gathering and you're eating a lot of protein, and you put on muscle mass and it benefits you in your hunting and keeping your family alive and passing on your genes and making children, and then poor food availability comes along, the times are hard, you lose muscle mass, you're eating less protein, your muscle shrinks in size. It would benefit you to then, when food availability gets better again, to be able to put that muscle mass on much, much quicker and at a faster rate, so that you can then get back to where you were previously in a seasonal manner. And that's why he reckons mammalian muscle fibers here, so we're talking mammals, not just humans here, have that as a mechanism of memory. And that's why it would make sense. And the interesting thing is that this never used to be a concept. Muscle memory never used to be a concept where, in the sense that myonuclei could decrease. The idea used to be that for muscle growth, myonuclei increase, and then when the muscle atrophies and gets smaller, the nuclei actually selectively degraded and disappear. Right. But that's been proven that was actually not fully true, because what they would do is they'd look at muscle fiber that was getting smaller and atrophying, and they'd break it down and they'd find markers of nuclear, so DNA degradation, and they say, the DNA is getting degraded, therefore the myonuclei are getting degraded. But they were looking at the muscle as a whole and not the fiber. And there's multiple nuclear types inside the muscle fiber. There's the myonuclei, there's the satellite cell nuclei, there's the nuclei of white blood cells in there, there's the nuclei that are involved in making capillaries and the blood supply. And so the capillaries and blood supply, the white blood cells and the satellite cells, they may decline and they may get selectively degraded. But it seems the nuclei that are inside that fiber, inside that membrane, are actually selectively protected. And why would the cell be wanting to do that? Well, on an evolutionary scale, it would make sense to protect them so that you can then reuse them later down the line to be better on a seasonal scale, to pass on ultimately more offspring, because your fitness and your ability to find food and procreate would be more.
Mm-hmm. Really fascinating.
And you could say the same thing with the fat cells. I mean, think about it same thing, seasonal food availability. It makes sense that you would put on a lot of fat quickly, and then if those fat cells just disappeared and then you were in a seriously dire time, and then you had more food availability, it'd be better just to refill more pre-existing fat cells that then help you survive. So
absolutely,
you got to think about it in terms of evolution.
Yeah, I think when we think about where we came from, and we haven't, I guess, that in terms of the lifespan of a human being on Earth, it's not been that long that we've had a completely different setup before we were kind of in the Paleolithic era or whatever. So we're still kind of, our genetic makeup is still very much dominated by that. We haven't evolved past that, even though we might not need this seasonal advantage now. It very much could be making up the background. So I think that's incredibly interesting. It makes a lot of just inherent sense. And I guess your study is going to help move us forward in terms of hypothesizing those sort of things, which I think is incredibly interesting. I don't know if you guys are kind of looking into it or you've thought about it much, but something that kind of alongside obviously kind of donating myonuclei and things and hypertrophy, I don't know if hyperplasia and the theory behind kind of, so the listeners understand, kind of increasing muscle cell number rather than hypertrophying and increasing muscle cell size. I don't know if this is something you've kind of got any thoughts about or have looked into at all.
Yes. So there's so the idea here is obviously muscle fiber hypertrophy. So your pre-existing muscle fibers that you have increase in cylindrical size, and that's the muscle growth, and you're not gaining new muscle fibers, and that would be muscle fiber hyperplasia. Muscle fiber hyperplasia has been observed in animal models under the cases of extreme loading. And it seems to do with where you actually damage your muscle fibers and cause them to split. And during that process of splitting, you actually then end up with two muscle fibers. And therefore, if you have two muscle fibers, you now have the ability to have two sets of fibers with their own sets of nuclei and more muscle fibers. You're going to have a bigger muscle. There was a very interesting paper. I think it was published at the very, very end of 2018. So It should be available by now, and it was called Muscle Fiber Splitting is a Physiological Response to Extreme Loading in Animals, published by Kevin Murak, and that's under the John McCarthy's group at the University of Kentucky. And essentially they're saying that in some of the studies that have taken place on powerlifters and steroid-using powerlifters and bodybuilders and steroid-using bodybuilders, both naturally and enhanced, split fibers are observed. And it would make sense that, and some of these guys are like world record level guys, like seriously strong guys. And so some of those authors that have written those papers and say that muscle fiber splitting may be occurring in these people because they're exposing themselves to extreme loads consistently for years and years and years. And maybe that might be a reason as to why muscle hyperplasia could be occurring in some very rare instances in these people, but there's not been any direct evidence of it occurring in humans as far as I know. But there are some tantalating sort of glimpses that it might be happening, but it's a very, very difficult thing to measure in humans especially, and so we can't specifically look at that. But what we can look at is if we can find any split or damaged fibers. And it's actually quite interesting that once you start to learn more about the microscopy work that goes into these kinds of things, you can see a split fiber is characterized by having its nucleus inside the center of the fiber. And so if you see fibers that have that as a characteristic as a split fiber, and maybe down the line that could divide and you could have two new muscle fibers from that. But you'd have to monitor that in a person through a time course, which is then incredibly difficult to do. And so that as a research question is something that needs to be investigated much further.
No, totally. It's just interesting because it obviously plays into a little bit like whether or not steroids have any role in play, like hyperplasia potentially and things like this. And to circle back to kind of obviously talked about people on steroids, like are there certain ones you're looking for in that kind of for people to be eligible? Do they need to be taking a certain dose, or what is the kind of prescription there?
Just above physiological amounts. So they are self-declaring that they are taking more and their testosterone is going above the physiological level, just so that we can basically scientifically kind of guarantee that androgen receptor steroid binding is happening at a much higher rate than in the other guys. And that's another difficulty with this study, is that it's highly likely that everyone's going to be on a different steroid cycle. Everyone's taking steroids for different periods of time. Everyone's taking stuff from different places. Whether or not it's truly what they think it is, we don't know. And so there's differences there. And the interesting thing is that all of these steroids interact slightly differently with the androgen receptor, and their ability to bind to the androgen receptor, that affinity is more in some steroids than in others. And that's where you can say that some anabolic steroids are more potent than others in terms of their ability to build muscle. And along with that, there can also be differences in how the anabolic steroid binds to the androgen receptor in other cell types. So how does it bind to the androgen receptor in the prostate? How does it bind into it in other cell types? And that's why some anabolic steroids are regarding has more anabolic effects and less androgenic or masculinizing effects, which is good because it's been impossible for anyone to fully disassociate the anabolic to the androgenic effects of these drugs, because the androgen receptor is the same in every single cell type in the human body. But what is different inside the cell types is all the other different, the cellular milieu, the cellular background is different in these cell types. And this is how Sarms work, because the idea with Sarms is they're selective androgen receptor modulators, and they were designed with trying to fully disassociate the anabolic to androgenic effects that were observed with people taking testosterone, because you can't give testosterone to an elderly woman who's suffering cancer muscle wasting, right? Or an elderly man, that could have potential adverse effects. So why not? And also if injections involved, that's just logistically a problem. So why not be able to give them a pill that they could just swallow that actually increases their muscle growth and actually causes less side effects? And they at Sarms have the ability to selectively bind to the androgen receptor in muscle, but not in, let's say, the prostate, which is commonly the one investigated. And the way it does that is, as I was explaining before, when these drugs bind to the androgen receptor inside the cell, and then they move into the nucleus of the cell, they then want to bind to these areas of DNA called androgen response elements. And you want that to happen in muscle because you want muscle growth to occur. That is the first step. After that, you then make the transcripts that then result in the proteins being made. But you don't want that to happen inside the prostate. And the thing is that there's a whole host of cofactors that have to bind to the androgen receptor to help it bind to the DNA, or to DNA, like full stop. And those cofactors are actually different in different cell types. So the cofactors inside the prostate are different to the cofactors inside the muscle. And so if the SARM, when it binds to the androgen receptor in the muscle, is designed in a way that it will attract the muscle cofactors, then that's good because it will bind to muscle DNA and then cause muscle growth. But if it, when it gets to the prostate and it binds to the androgen receptor in the prostate and then it's making its way to the nucleus, if it then doesn't bind cofactor very strongly because it doesn't interact very well with prostate cofactors, then it won't bind to prostate DNA and won't elicit an adverse effect. And that's how SARMs ultimately work. And that is being further investigated. They haven't been proven yet for human consumption, but and phase 3 clinical trials. But that's how they fundamentally work. And so dissociation is all to do with how the androgen receptor, once bound to the drug, binds to the DNA. And the idea is that if you can design the drug so that it attracts muscle cofactors, when it's inside muscle, it will bind to muscle DNA and give you the response you want. But if it's in the prostate and it does not bind prostate cofactors, then it won't bind to prostate DNA and you won't get the adverse side effects, which is what people are looking for.
Mm-hmm. And to come back to the kind of the study and what you're trying to observe, and in terms of kind of the muscle memory effect from those who have taken kind of the steroids beforehand and then coming off it, what's your hypothesis? Is there kind of a thought that they might have somewhat of an inherent advantage in future, like you kind of described, kind of build muscle in your younger years so you have more later, and then likewise someone who's on the kind of gear, they're going to build even more muscles, they might have an inherent advantage in future years? Is your hypothesis that that would be the case?
Based on the cellular models we've seen in mice, yes. But as I was saying, if we're talking about individual people here, even if they go on a cycle and then they go off, it's like, well, how disciplined are they afterwards to adhere to training and nutrition? So that, I mean, straight away you're removing an anabolic stimulus of enhanced rates of muscle protein synthesis. Like that's going down. So the question then is are you then still, if you've got more myonuclei, are you still able to activate them all to the extent that they were activated when you were taking steroids? We don't know the answer to that question. And even if you were fully diligent with your training and nutrition, are you still able to make the most of the extra myonuclei that you have? And we don't know that until the study is done, where people are exposed and then removed and then exposed to training without the drugs being in the system. And I mean, I've read one thing in Gunderson's stuff where he's saying basically like, even there, it might be that you might not fully be able to make the most of those myonuclei to have a truly enhanced rate of muscle growth 5, 10, 15, 20 years down the line. Okay. Because your training and nutrition's got to be to a certain level, and you might, even if you're really diligent, never be able to get to that point. But in theory, you would actually recover faster because your protein turnover should be better. So if you're damaging your muscle fibers and actually damaging those proteins and tearing them, if there's more nuclei in there, the propensity to recover might be better. But again, that's theory. Like, to actually what extent, you don't know. You'd have to sort of do protein turnover studies in people that have more myonuclei than don't, and then make a conclusion from that. But in that is something that has been discussed as one potential advantage. So it could be that your propensity for muscle growth is enhanced, but is that actually actionally possible in the real world? We don't know.
It's interesting, I think, because a lot of the listeners here will be like physique athletes potentially, and some federations, I know in the UK, they have like a drug-free rule. Like if you've ever taken anything that's a banned substance, like you are not able to compete. And I know there's one called the BNBF, which is a big UK bodybuilding federation, and they, I believe, still have a 7-year drug-free rule. So it's kind of interesting to hear whether or not those people that maybe, I don't know, 8 years ago were on anabolic steroids, they gained all this muscle, then didn't, and maybe do they have that inherent advantage? Is it fair for them to compete? I don't know if you have any thoughts initially on that. Yeah,
I mean, that's where I'm saying it's very difficult to know, and you can see why they decide to do that. But truly, in real life, we don't have the long-term studies to really know how much of an advantage does that person have. Like, you might have someone, say, who competes in, untested bodybuilding in their early 20s, and then in drug-free bodybuilding in their mid-30s, and it's kind of like, do you have a comparable person who competed in drug-free bodybuilding in the early 20s and is still a drug-free bodybuilder in their mid-30s? And if everything was kept the same and that was the only difference between them, a very difficult thing to do.
Yeah.
Is that person on stage in their mid-30s actually bigger? We don't know. And in theory, that could happen, but all the other things have got to be in place for that to actually be witnessed and to occur. But I mean, drug testing with anabolic steroids, though, is 6 years, 7 years ago, that you're never going to know whether or not somebody has used it. And so hence the polygraphs and the things like that, are the best thing you've got, especially when you're talking years and years ago. It's interesting, though, because things like nandrolone decanoate are detected in the urine and the picogram amount. So that's 1 × 10⁻12. So that's 1,000 times smaller than a billionth of a gram. And so that's a tiny, tiny amount. And it's been observed in studies where they've given people that drug that up to nearly 2 years later, you can actually find traces of nandrolone. So, that's, as far as I know, the longest detection window that you've got. And even then, it actually is realized now in anti-doping that people's metabolism and the excretion rates of drugs is very individual. Yeah. And there is population averages, but then athletes are not the population average, and there's differences in how they're excreted, and so it could be even more or even less than that.
Yeah, it's really interesting in that people could be, I guess people should realize this, that you can be clever with the way you do things, and if you've got someone who's working with you who knows their stuff in terms of all of this, you could pass the blood and the kind of piss tests and things like this, and it would just be the polygraphs that are kind of whether or not you can pass that or not. And I don't know how valid they are. They're obviously not used to a large extent in many cases in like the government and things, because there isn't a whole lot of validity to them. So I think it's good that people are aware that obviously these sports try and be as clean as possible, but there is still an inherent risk that this sort of thing could be going on. Yeah,
of course. It is something that, and then what do you do as a federation and then trying to be fully drug free? Yeah. When you could have people 5-10 years ago, there's nothing you can do. You could just do your best and hope that people will adhere to that.
It's kind of the test that as more of a deterrent than anything else. Yes. And then, for natural bodybuilding, for example, it's not like you're winning huge bucks or anything. It shouldn't be something that people are trying to necessarily cheat the system. Something I definitely wanted to finish off with you, Alex, before we kind of run out of time, because I think it's super important. I think it's really relevant to our kind of audience. You spoke a little bit with Danny about kind of the anabolic use and the potential side effects and the kind of bad things that happen. You kind of spoke about it a little bit here in terms of like why Sarms are becoming more popular. And within the UK, because of the fact it's legal to be able to use them and it's just selling them that's illegal, it's kind of growing massively. I know you kind of looked into this yourself. I'd love to hear kind of some words about that and kind of your thoughts and feelings.
Yeah, and I'm really happy you brought up harm reduction, because it's so important that in the United Kingdom, they're a Class C drug, which means possession is not criminalised. So you can have the drugs, but manufacturing, buying and selling is criminalised. But that's not a priority of the police force to find people really who are selling on a small scale anabolic steroids. Like, let's be real here. And the truth is, if you look at the statistics, the increase of anabolic steroid usage amongst young men in this country in their early to mid 20s is up, and people are trying to wonder why that's the case. Maybe it's people feeling pressured by social media. Maybe it's fitness in general being more popular, bodybuilding being more popular than it ever was, and people becoming a bit more savvy that actually your elite IFBB pros are not just on chicken and broccoli, and which maybe in the days of the magazines, and especially in the United States where they are controlled substances, was not discussed. Whereas now, at the age of social media, that kind of thing is out there. And I mean, people have just got to make a very, very educated decision if they want to take that path. And and harm reduction is something that I think everyone in the fitness industry should be talking about, because we want to make sure you can do the best to safeguard people, because it's their life, it's their body, it's their choice, and you can do whatever you like. But the best option is that you're informed about the decisions that you decide to make, because anabolic steroids are no joke. They will shut down your internal testosterone if you take enough of them for a long enough period of time, which is highly likely what people are going to do anyway. So you're going to have an exit plan on how you're going to stop using these drugs. Are you going to blast and cruise, where you're going to take a high dosage and then cut down to testosterone replacement dosage and then go to a high dosage again? Have you got all of the accessory drugs that are required for that? Or are you going to cycle off, and have you got the accessory drugs to conduct a true post-cycle therapy? And do you understand that the safest way to do that? Dr. Dean St. Mart has a very, very good YouTube video on how correctly to conduct a post-cycle therapy. And, we know that and this has been published in surveys from the United States, that most people that start taking anabolic steroids, about 1/3 of them will continue that for the duration of their life. And again, Kanayama and Harrison Pope, the guys that have conducted those surveys, say the main reason for that is that towards the end of a steroid cycle, people have obviously achieved some goals. They've got bigger, which is exactly what they're taking them for. And then either they psychologically cannot deal with the idea of losing their gains, and physiologically, when they go off steroids and their internal testosterone is very, very low, if they don't do a correct post-cycle therapy to manage and mitigate that, hormonally, because of that environment, they feel crap, and they might then go back to usage because of that reason. And so they say you can end up in this situation where you sort of continuously using steroids, and also at the same time, they think that that's one reason, where basically people can't get off. Mm-hmm. So that's one thing, is that if you've got to be educated on your exit plan for the drugs, and also if you are blasting and cruising, because your internal testosterone production is shut down, if you're planning on doing that for a number of years, you have to weigh up, am I going to be able to kickstart my internal testosterone production again? Because spermatogenesis and the ability to make sperm is reduced while you're on steroids, because your testes mass will decrease in size in most people. Obviously, everything we're saying here is in most people. This is like the average case scenario. You might be the outlier where it's not as bad as another person, and you might be that poor person that goes on one steroid cycle and totally shut down their testosterone and struggles to ever kickstart it again. And there is such a condition as anabolic steroid induced hypogonadism, right? AISH, and that is being described by various people who are involved in designing PCTs in the scientific literature. And so you've got to be conscious of that, because this could affect your ability to have children when you're older. And so you've got to make that life choice now and be mature enough to know that. And there's nothing wrong if you want to take anabolic steroids. That's your personal decision, mm-hmm. and you're going to be a diligent user, and you know your exit plan, as we just discussed. Then that's your life and your choice. And Mike Isotel has spoken that there is ways to take these drugs safely. Yeah. And the other main things that you've really got to think about is then just monitoring your health while you take them, and that should just be an absolute no-brainer, because anabolic steroids will increase your red blood cell count. They'll increase your hematocrit, and if your hematocrit is highly elevated through time, that increases the likelihood that your blood is very thick, and that could cause kidney damage if that is in that case scenario is happening for many, many months on end and for years on end, and you have no idea that that's occurring. Anabolic steroids can increase your bad cholesterol and decrease your good cholesterol, and that is a risk marker for cardiovascular disease. And if that is unmonitored for months, years, decades, then you could have set yourself up in an environment to have an increased risk of heart disease. And so you've got to monitor your blood pressure as well, because the same thing, your blood pressure can get elevated, and again, that could cause damage to your kidneys. You've got to monitor your red blood cell count. You've got to monitor your cholesterol levels. You've got to monitor your liver enzymes, especially if you're taking oral steroids, because you have to be aware of are your liver enzymes, which are a marker of liver health, elevated for large significant periods of time? If they are, then you're increasing the likelihood that you have liver damage. But everything that I've just described there, that increases in blood pressure, the abnormal and disadvantageous variations in blood cholesterol, the elevation of liver enzymes, all of them are relatively short-term side effects that do dissipate and do go away. And obviously it's a case of the more you take and the longer you take it, the higher the likelihood that those things go in an adverse direction. And if that's maintained through time, the higher likelihood that you can have a morbidity associated with it, cardiovascular disease, kidney damage, liver damage. But if you are a diligent user and you are monitoring your health and you are making sure that those things are not way off scale and are more in range, and are using low to moderate dosages for not really long extensive periods of times, and you're just sensible about it, and that's why speaking about this harm reduction is the most important thing, then it just decreases the likelihood that you're going to have adverse side effects later down the line that maybe you didn't know were even theoretically possible, you didn't feel were happening to you, but you didn't get your blood mark and your blood work done to check that. And this is expensive. Like, you're going to have to buy everything that's involved in taking them, the injection materials, the drugs themselves, the accessory drugs to go off them, and get your blood work measured consistently. So you've got to make sure you're up for the long road in that regard if it's something that you're really interested in doing. And just make that personal choice yourself, and be diligent and sensible about it. And that's why harm reduction is the best way to go about this discussion. Mm-hmm. Because, as I said, a lot of those side effects are transient, but if apart from the shutdown of testosterone, that could potentially be permanent. But, the elevations of blood pressure, the elevations in liver enzymes, the adverse cholesterol profile, all of that can go away if you were to stop taking them or to take less. And so if you're mature enough that psychologically you can make that decision in that moment, which takes a mature person to be able to do that. You might be making the best gains of your life, and strength is through the ceiling, and muscle growth is just something the best you've ever had, and the pumps are out of this world, and you might feel like Superman. But if your health markers are going down the drain and you're not wise enough to say, actually, I've got to be careful now, maybe I should just take a little break and then go again, or at least take some things to try and reduce my blood pressure, reduce my cholesterol profile, reduce my liver enzymes, maybe stop taking oral steroids, etc., things along those lines, then you could be regarded as a safe user. And it's better, I think, to try and, if people are going to decide to use, to try and promote safe usage instead of just saying, no, don't use it at all, because that's not the society we're living in at the moment. It's becoming more common. So it'd be better to say, if you are using it, try and just be a diligent and safe user for your own personal health. And that's going to benefit you in the long run and ultimately make you healthier, and in the long term, make you retain and have better gains. So that's the message that I think has got to be talked about to safeguard as many people as possible.
I couldn't agree more. I think that message you said there is incredible. I think it just goes to show how much thought that needs to be put into having taken them before you take them, the kind of the logistics behind that, how much investment there's going to be, and then obviously time away from them, and then really knowing yourself. Because I think, unfortunately, a lot of bodybuilders are that obsessive personality who do not like to pull back and do like to push harder. And if you're that person and steroids sound like a risk to you, potentially it's not worth the reward. So I absolutely love that message, Alex.
And that's why, you might, there's coaches out there who know about this stuff, and if you think I'm the kind of person where I'm in that best cycle, gains are through the ceiling, but my health markers are bad, and then your coach should help you identify that, and then so that actually, look, let's take a step back. We'll get back to this point and do even better once you're fully healthy with your blood work. And you then sort of need to be able to realize that and take that step back. And I think that conversation has to be had just because of the increase we're seeing in usage.
Absolutely. And I mean, the same is in kind of with naturals, and that people always want to continue to push the diet. They want to push their training harder. And as a coach, you have to pull them back and save them from themselves. So I think, especially for obviously like new time users, like if they're going down that route, then a coach is a great idea. So no, fantastic, Alex. I've absolutely loved this discussion. I think we've given some tremendous value. Before I kind of close off, I definitely want to kind of again reiterate kind of what you're looking for, where people can find you, and how people can share your message and get this research going.
So I have, as I said, public Facebook post. You can just search for Alex KT Muscle Memory and you should find it. Otherwise, Mike Israetel has recently shared it on his Facebook page. You can find it there to be shared. I also have an Instagram post, Alex_K_T, and that describes, and that has the advert for our study and what we're looking for. And specifically, it's males aged 20 to 35. Who are, number one, sedentary controls that don't lift any weights. And actually be surprised, I'm actually finding it quite hard to find those people because
everyone lifts now.
And also people that lift are interested in what we're trying to find, whereas you've got to persuade those other people. So if you have friends and colleagues, or even just sharing my advert can help get the message out. So sedentary controls that don't lift weights, guys that lift weights regularly, ideally, and have been for 8 hours a week consistently, and guys who are in training hard as well, just like that, but they're on anabolic steroids. and they've just finished or planning on just finishing taking anabolic steroids, and they're going to cycle off. And so that when they come to the labs, they've either just finished or maybe a few weeks, maybe even a month ago finished, and then we sample them, and then 20 weeks later they return to the lab so they can see how much muscle mass they've lost through time. And then guys that used anabolics 2 to 4 years ago but since then have been drug free or on a true genuine TRT dosage proven with blood work. And you come to the laboratories, the University of Brighton laboratories in Eastbourne, postcodes BN27SN. Full travel expenses will reimbursed, potentially from anybody in Europe. We can have that discussion if you're interested. My email, as Steve said, will be in the show notes. That's the best place to get a hold of me. Or if you're Alex_K_T on Instagram, have a chat. And if you're eligible and seriously keen in taking part, then eventually we'll speak over the phone and then plan your trip. Saturday 16th, Sunday the 17th of March, Saturday 13th, Sunday 14th, Monday 15th of April, we have sampling, and sampling will continue throughout the whole duration of 2019. So even if you're listening to this way after those dates, doesn't matter, and you want to take part, still get in touch, and I'll just let you know new dates when they're announced. And as a participant, we basically do paperwork. We'd collect your blood, urine, and saliva. You'd have your muscle mass measured with a bioimpedance scale, research grade 1, research grade skin fold calipers for your body fat, and then a bod pod. And then we would conduct a muscle biopsy of the trapezius. That's done obviously with full medical professionals. And everyone is obviously kept anonymous. And that's who I'm looking for. So thank you very much, Steve. I really appreciate it. I had a great conversation with you. And, please do reach out if you're interested. Anyone that's listening, I'd love to hear from you.
Amazing. Yeah, I mean, likewise, amazing conversation. And I'll make sure that we link that up all in the description box below so it's really clear for people, and make any adjustments so we can get the message shared as best as possible. So again, thank you, Alex, and thank you everyone for listening, and please do share the message. Cheers, guys.